Hello, everyone, and welcome to our chat about cells. And we're specifically going to be looking more at prokaryotic cells. But I want to start this conversation on what a cell is. So, a cell is the basic unit of life. All living things are made up of cells, and cells come from pre existing cells. That's the cell theory. Now, we divide cells into two basic categories. We have our prokaryotic cells and our eukaryotic cells. So, prokaryotic cells are unicellular. That means the entire entity is one single cell. And our prokaryotic cells are unicellular. And if you look at the picture on the left, you're going to see that they really lack internal organization. Everything inside that cell is just kind of thrown in there, hanging out together. We do have all the genetic material kind of condensed in one area. We refer to that as the nucleoid, but everything else is really just hanging out in the cytosol. So prokaryotes in general tend to be smaller and more structurally simple compared to the eukaryotic cell, which you see here on the right. So eukaryotic cells tend to be larger and they are more structurally complex because, as you can see by looking at this, eukaryotic cells have two identifying features. A nucleus, it is a membrane-bound organelle that contains the cell's genetic material. Also inside the eukaryotic cell are organelles, and organelle translates into tiny organ. So these organelles are basically the organs of the cell. And just like in our body, our organs have different shapes, they have different jobs, they're located at different positions within the body. These tiny organs or organelles are the same. They have their own structure, which relates to their own function. They have their own place within the cell, and they each have their own environment that allows them to do whatever their job is. Now, eukaryotic organisms, which are made up of these eukaryotic cells, they can be multicellular or they can be unicellular as well. For instance, some yeast are unicellular eukaryotic cells. So let's zoom in real quick on just prokaryotic cells because that's what the bulk of this class is going to focus on. So as I mentioned before, prokaryotic cells are unicellular. The entire bacteria shown here is one single cell. It is typically much smaller than a eukaryotic cell, and it lacks that internal structure. There are no organelles, there is no nucleus inside this cell. And we're going to go into more detail on the external and internal components of prokaryotic cells in later videos. So for now, I just want you to just take a look and kind of admire how it is simple yet kind of complex, right? There are lots of components to these prokaryotic cells. And like I said, we're going to dive deeper into those soon. So one thing about bacterial cells, and which are prokaryotes, but again, most of this class focuses on bacteria, are going to be their morphologies or their overall shape. And so prokaryotic cells have a few basic cell morphologies that are listed in this table. I've crossed one out because we're just not really going to talk about it in this course, but that it does exist. So in this class, we're actually going to focus on three different morphologies or shapes, and that is going to be coccus or cocci if we're talking about multiple cells. And these are going to be our predominantly spherical shaped cells. Bacillus or bacilli, if we're being plural, are more rod-shaped cells. Then the other one we're really going to talk about are the spirochetes, and these are our more thin spiral-shaped cells. Now, spirillum is another spirally morphology, but if you compare the two pictures, you'll notice that the spirillums tend to be a bit thicker and they're less wavy than the spirochetes. Another one that does exist is vibrio, which are more like curved or comma-shaped cells. Now, in this course, as I'm trying to help you become familiar with the terminology, I may say a bacillus or rod-shaped cell. But when you are talking in the course, when you're completing assessments, you should always use the microbiology term. So on a knowledge check, if I show you a picture of a cell and I ask for its morphology, you'll say bacillus, not rod-shaped. Okay, we're always going to use micro terminology. So again, morphology is the shape of the cell. The other thing we will be looking at, especially in lab, are the bacterial cell arrangements. And so the different arrangements that are possible are going to depend on the morphology of the cell. And that's because bacteria divide in different ways. And the plane of division is going to determine the bacterial cell arrangement. And those planes of division depend on the shape or morphology of the cell. And so this is going to kind of make sense because circles, right, are symmetrical in many different ways. Okay, and so, right, it's symmetrical vertically and horizontally and diagonally. And that's all going to come into play when we talk about the different arrangements that a coccus cell can have. Okay, so when it's talking about cellular arrangements, we're looking at how these cells arrange themselves as they divide. Okay, and so as one cell divides into two, kind of what's happening there. So for both coccus and bacillus cells, they can exist in a single arrangement. That means as a cell divides, it goes and it hangs out on its own. Both coccus and bacillus cells can exist in a diplo arrangement. That means they are arranged as pairs. Okay, notice they're going to be end to end. Now, here's where I need you to see what's happening here with our bacillus cells. Okay, notice they're arranged end to end, and this is and not stacked on top of each other. And this is important because if we have a bacillus cell, okay, it cannot divide long ways. Okay, so it's never going to divide and then like stack like that. That's not going to happen. The cell cannot divide that way. Okay, so if you kind of put your hands together in like an oval shape and you kind of divide and pinch them out, that's how they're going to divide. Okay, so these bacillus cells are only going to be able to attach end to end like that. Okay, so when you have two bacillus cells arranged that way due to their plane of division, we call that diplo. The next one we're going to talk about are tetrad. And this means four cells. It's a group of four cells arranged kind of like a square. And you'll notice that you only see this with our coccus cells, and that's because of their symmetry. Okay. The coccus cells are able to divide both essentially long ways and short ways, right? Because it's the same thing. And so they are able to essentially like stack on top of each other. So if we're referring to a tetrad arrangement, you know that the morphology of the cell must be coccus. The next arrangement that we're going to talk about is staphylo. And the more common word that we use is a cluster. It's a cluster of cells. And so I've made a little cluster here on the slide. Now, they don't all have to arrange exactly, okay, like that. They're basically, and I'm sorry, they're not overlapping in that capacity. That's just my poor drawing skills. Okay, but they're hanging out as a cluster or group of cells. And this again is because of the symmetrical nature of coccus cells. And so staphylo is the arrangement for that. Strepto is an arrangement that we can observe with both coccus cells and bacillus cells. And this is when those cells arrange end to end to form like a chain. Okay, and so because of how bacillus cells divide, they can form the strepto arrangement. Okay, but they cannot form staphylo because they cannot divide in such a way that allows them to stack like that. Okay, again, that won't happen. So you will not see any bacillus cells with the staphylo arrangement, but they can form strepto. All right, so what I have here on the screen is the arrangement terminology. But when we are talking about the morphology and arrangement of cells, we merge them together in a specific way. Okay, and that's what you're seeing down here at the bottom. So if we were looking at these cells right here, and I say, what's the morphology and arrangement of these cells? You actually first address the arrangement. They're end to end, right? So our micro term is strepto. And then you would address the morphology, which is coccus. So it's streptococcus or streptococci. I personally am not picky. Okay, but notice it's written as one word, and we have the arrangement first, and then we address the morphology. Okay, but it's written together. So following that pattern, if I asked you to give me the morphology and arrangement of these cells, okay, you would first address the arrangement, so diplo, and then the morphology, which is bacillus. Okay, again, or diplobacilli. Now, the only time you do not write it as one word is when the arrangement is single. You would say that this one is single coccus and this one is single bacilli. I'm sorry, single bacillus. And those are two separate words. Okay? And again, on assignments and in conversation in class, we're going to be using our micro terms. So if I asked you, what is the morphology and arrangement of the cells that I circled in purple, you would not say it's a cluster of round cells. Okay, you would say staphylococcus. So staphylo is the micro arrangement term, and coccus would be the microbiology term for the morphology. Okay, so we're always using our micro terms. All right, y'all, that is it for this video. Let me know if you have any questions.